Now I try to explain you these activities. So looking at plant cell through a microscope, that is we will see. You will need microscope. There is the microscope. So microscope slide. So this is the microscope slide. So this is the microscope slide. Cover slip. That is the cover slip. So inside the square shape mirror is there. This is the cover slip. Onion bulb. So you see here, there is the onion bulb. Here is a onion bulb. So twicer, forceps. So here there is no forceps. Sharp knife. There is a sharp knife. The small container of water. So there is the small container of water. Then everything I took. So first, so collect a small piece of onion, cut out a piece of about 1 centimeter square. Already I cut it the 1 centimeter square. Okay. Use dropper pipette to put a small drop of water into the middle of a clean microscope slide. So middle of clean microscope slide. So this is the microscope. So middle microscope, then I put one drop of water. Just middle microscope, I drop. Okay. That is, I drop one drop water. So use the dropper pipette to put a small drop of water into the middle of clean microscope slide. I drop it water here. So next one, very carefully peel the thin layer from the inside of your piece of onion. So already I took it this layer inside the onion. So this is the layer. Okay. Gently push the layer into drop of water on the slide. Spread it out flat as you can. So I take it this one. Then I put it here. Then I spread likewise. Okay, then I spread likewise. This is the onion. So next one, fourth point, it is finished. So next one, gently push the layer into the drop of water on slide. Spread it out as flat as you can. So I spread it. So collect very thin piece of glass called as a cover slip. So that is the cover slip. Then I take one. Okay, this is, I take cover slip. Take care, cover slip break very easily. Gently lower the cover slip over your piece of onion on this side. So we will keep it over the piece of onion, this one. Okay, I keep it that one. It is also okay. The cover slip I kept over the onion. Try not to air bubbles under the cover slip. It does not create any air bubbles here. When you keep here the air bubbles, it is not be created. So you keep likewise. So turn the objective lens on the microscope until the smallest one over the hole in the stage. Turn the objective lenses. So objective lenses means here. Here I keep it there. There is a one glass mirror is there here. Then center of that mirror, I keep it this one. Okay, keep it here. So after that, look down to the eyepiece. Slowly turn the focusing knob and move the lens away from the slide. Stop when the piece of onion comes into the focus. So now I adjust the mirror everything. So mirror, so it is I adjust. Then I am looking here. I am looking here. Just I am seeing here. And now you will get cell structures, but I don't know how to you see the cell structures here. Then how do you focus? Then I don't know. So okay, now you will get it the cell structure when you adjust this adjusting knob. Okay, in this way you try, you can get it a plant cell structure. Thank you.